introduce Dr. Rosina Mahroof. Actually, I learned about her because Xiaodan, my student, she was doing the stored product research, and I realized, oh, Rosanna also does this research. So she now is a professor of biology in the 2 departments. You can see from the slides, basically. So she just mentioned that the second one, Department of Biological, no, the first one, Department of Agriculture and Public Service is a new one. So she's the acting chair for this department. She got her bachelor degree in agriculture from University of Peradeniya, Sri Lanka. Then a master degree in forest entomology from University of Edinburgh, UK. Then she earned the PhD degree in stored product pest research in Kansas State University. After that, she spent 2 years in Oklahoma State University as a postdoc. Dr. Mahroof published over 65 articles and presented over 168 presentations. She serves as the stored products pest section, a subject editor for Journal of Economic Entomology and the Journal of Agricultural and Urban Entomology. She has received 36 awards and recognitions honoring her work, including the Governor's Award for Excellence in Scientific Research in 2018, and also the Fulbright Senior Scholar to Sri Lanka from 2019 to 2020. She received close to $13 million worth of competitive funding as PI, co-PI, or collaborators. So with that, I welcome Rosanna for presenting the seminar today.
Thank you so much. And first of all, it is my honor and it's a great pleasure to give a seminar to the Rutgers University Entomology Department. I really want to appreciate Changlu as well, to encouraging me to inviting me to give a talk. So I wanted to recognize that, and along with Dr. Brad, I think it's Brad Sten. She communicated and kept me abreast of all, the updates. So it is indeed, I'm just going to present my talk. I hope everybody can see my slides here. And I'm sure that not many of you are not working in the aspect of stored product entomology, so except probably Dr. Changlu, who has some research going on at this time or done some work in the past. So I chose that title as a very general title as stored product pest management in a changing world. Just to pinpoint some of my challenges experienced across the board, as well as what are some accomplishment throughout that time. Just to give a all right. So just a concept of stored product entomology, I would really like to say that it is a man-made problem. It's just we created this monster during the civilization. Since civilization, we started to stockpiling our food as human and invited all these feral insects, which were basically living in forest areas or feral environment, and into this stockpile food, which coming into the habitat of the human civilization. So the losses, basically, if you really look in the United States, is about 5 to 12%. I'm just citing here for the stored durable commodities. These losses are very much high for a perishable commodity, and that's not part of my work. And this is in the United States, but there are other countries, developing countries, the losses can account for about 60%, something where 50 to 60% of the final harvest. The two majority of the group of insects which are causing this problem belong to the beetles and moths. And there are two types of damages. Predominantly, there are insects which are invading inside of the grain, and they are feeding all the content of the endosperm and everything germ, and the content of the seed, that's causing a direct loss. Or there are insects which are basically feeding from outside of the grain or value-added product or damaged grain. And sometimes, value-added product means like flour or cereal or any other dried fruits and nuts. And they can also cause direct losses. At the same time, indirect losses are caused by contamination of food by webbing materials and fecal matter, molted skin. And stored products storage structures are basically also some damages are caused to the structures. So since this like location factor, right? Like buying a real estate, the location is the key for this insect. So occurrence of these insects, they are along the supply chain, starting from farmers' bin, go to the elevators and country elevators or terminal elevators. From there, it is transported to various mechanisms like trucks, river barges, ships, and trains, coming into food processing. And from there, it goes into warehousing and to your kitchen finally. So this is basically the farm to fork supply chain. And the insects can occur from anywhere from farmer's bin to the kitchen. So actually, if you look at the stored product pest management, the caveat is the problem is persistent. Regardless of, we may come up with numerous different control tactics and all the education, training, and outreach, everything, still the problem is persistent in the food industry. So what it happen is basically when there's a problem encountered, that's when it's not proactive, it's basically reactive. So often they try to make a quick fix and then not thinking about the long-term solutions and rarely learning from the mistake. So this can lead to frequent occurrence of insect infestation, leading to recalls of the product from the market, can lead to cost millions of dollars for the industry. And now we are living in an ever-connected world, so the bad news spreads so fast. So once a product is recalled, sometimes for example, if there's a breakfast cereal with a moth infestation that came on the news, then we would never go back to that brand again until we become more comfortable with that, right? So it's a lot of losses for the industry. So what are the challenges we are facing now in stored product pest management? So the major problem is the elimination of key pesticide from this chemical registry. Since 1996 FQPA, that is the Food Quality Protection Act, many of the chemicals, especially organophosphates, were eliminated, which were used heavily. And one of our crystal ball was the methyl bromide. Fumigation is solution for many problems in the stored product pest management. That was now completely eliminated due to the problem it causing in the ozone layer and completely phased out in United States. So we have some few tools existing, but again, those tools also have problem because insects are developing resistance to pesticide. And one of the tool is the phosphine fumigation, came very popular after methyl bromide. Now, many insects started to develop resistance for that. Then the behavioral trait of stored product insect. This is a very peculiar insect. They can successfully survive in man-made environments with tiny little of food like dust accumulating in cracks and crevices, and without any water, because they can survive about 6 to 8%, 9% of water content of the thing. And they are long living, highly reproductive. So this behavioral trait making them very successful in that environment. And on top of that, there's a greater consumer demand for pesticide-free food now. So all these are the current challenges we are facing, and we need to cater this in order to face this, eliminate the problems or the challenges. The pest management approach should be a very holistic approach. So to start with, rather than reactive, be proactive. So prevention is number one. Without waiting the pest problem to occur, we really have to prevent the problems which can occur in the future. How do we do that? So basically, we need to have establish an economic threshold, which is like very denominator for many pest management program, and it's mostly done by sampling and ID, and then identification, and then if needed, its control is done. But make sure that the sampling and control cost is really less than the market value of the product, and then bringing into different tactics together as an IPM method, incorporated with chemical and non-chemical tactics, and come up with a holistic approach. So with that, I really want to summarize my experience in stored product insect management, and I'm going to go along the supply chain and highlight all the past work that has been done. So starting in the farmer's bin, a lot of work has been done in the dispersal behavior of insects and determining their origins and feeding habits using some intrinsic biomarkers such as like the carbon and nitrogen isotope or trace elements. And from there, coming into transportation and processing industry, work has been done on physical control, which is basically the heat treatment for structures and trucks and trains, fumigation using ozone, and packaging methods such as exclusion. So then to warehousing and up to the pantry, the work in my past experience is in the host selection and biology of the insects, mass trapping, mating disruption, mating delays, and some other areas such as sanitation and bioplastic. So from there, I also have some experience in the international work. I have been part of a USAID grant for past 9 years in the Ethiopia Feed the Future program, and also some work with the initiated through my Fulbright program, the stored product research in Sri Lanka. Given this, I am given a 1 hour. I don't have time to cover almost all of these areas. So I only picked some area to talk today. But if you have any interest, please do visit with me or contact me to, I can discuss of the topics which I'm going to omit from the talk today. So let us start from the farm, right? So the farm, where the farmers been, one of the major problem with farm storage is the internal feeders. The lesser grain borer is a great pest in wheat, corn, and all kind of freshly harvested grains. Rhizoperta dominica, it's a Coleoptera bastricidae. So first of all, we wanted to determine the natal side of the Rhizoperta dominica using chemical and physical technique. So here is what I'm going to talk a little bit about. The looking around, we actually looked at the abundance and occurrence of this pest. So initial trapping done showed that very interesting data. Indeed, you expect to catch a lot of lesser grain borer in the grain storage environment. That is norm. However, we also caught lesser grain borers in habitats many, many miles away from the grain storage in open areas like grasslands and wooded areas. This really intrigued the question. Of course, there are alternative hosts or alternative populations existing apart from the grain storage. So because these are very strong flyers and they are occurring in distinctly different habitats, and if we are targeting our pest management only in this storage environment, we are omitting a larger population which can migrate into the grain and decolonize whatever the practices which eliminated before now become a problem again. So can we really define the movement and habitat of these insects? Can we look into their dispersal behavior? What are they eating as an adult food source? Can we look into the dietary history? Because we don't know what are their diet mixing. They are flying from one habitat to another. So in order to find answers for these questions, some objectives were developed. Specifically, we wanted to look into the dispersal distance of how far they can fly using mark release recapture studies. It's a very classical study. Also, we wanted to determine their, what they feed on it using some instinctive markers like carbon and nitrogen isotope and trace element marking. Again, due to time limitation, I'm just going to briefly discuss these two topics and going to eliminate the trace element profiling. So mark release recapture studies were done in two habitats, actually a forest habitat and a pasture open land. And basically what we did is we took wheat and we colored them using dyes of, green and orange for different habitats and introduced lesser grain borer into this and allowed them to infest the wheat. And then we actually custom built these grain bins. Those are smaller version of the farmer's bin, and then put them in those forest or open lands and put our infested grain inside of this, which are already marked. So if insects are basically flying away from that, they are going to carry the dye with them and actually go into mix with the feral population. And then we basically have to trap it. So we install traps and then we trapped. And just to share the results, in the first year, we found in the forest area, our percentage recovery of the mark beetle was really good, about 26%, closing to 5,177 insects. What is also interesting, you can see here the pattern flight behavior of the feral versus the marked insect, the green. They are almost following the same flight pattern, which is coinciding with the environmental conditions, and which basically we caught a lot of insects of feral, but about 26% of them were the marked. Same thing for the open side. But what is the conclusion out of this study was we placed our traps in 4 directions, east, south, west, and north, and different distances. And our traps are even from 2 kilometers from the original site of release, we caught insects. So they can, at least in this study, they can disperse even beyond 2 kilometers. And that is very interesting to note. Then to determine the carbon and nitrogen isotope pattern in order to find, basically this is the principle behind you are what you eat, right? So whatever we eat, that would be resembled in our body cells and tissues, and we can really look into delineate that history of feeding. And for this, we did laboratory versus field studies. In the laboratory, we selected the 3 hosts. One is acorn, just to resemble or assuming that in the feral insects or the forest wooded areas, they are feeding on these types of food. And then we selected, of course, the wheat and the corn. The reason here is very important, and I'm not going to go into details of plant physiology, but corn and wheat belong to a group known as C3 plant. You may recall your old plant physiology or botany where you might learn. They enrich a 12 carbon, and the body signatures or the carbon nitrogen isotope they have predominantly is 12. Corn belong to a C4 plant, which is enriching more of the 13 carbon dioxide. So the carbon isotope they have predominantly is the 13 carbon. So this is the basic for identifying the difference between what they are feeding on C3 plants or C4 plants. Now we went to field again, and then we did all this trapping work in the forest areas, open lands, and of course in grain environment, several different storage environments. And here the top graph shows our laboratory carbon, the nitrogen ratios, and the bottom graph is from the field. And of course, from field or different clusters are there, but they are very different areas from different localities. What is so beautiful in the laboratory is the C3 plants, you can see them, acorn and wheat clustered together here for their dietary history. And the C4 plant is very far away from the C3 plant. And in the field, we don't know what the insects we caught fed, but most of them fed on the C3 plant. Some of them fed on the C4 plant, and we can relate to that sites where they are, for example, the storage site or in a forest site. And what is so interesting also, some of them are mixing their diet. So some of them had fed both C3 and C4, so they had an intermediate isotope ratio. So that actually shows that they are basically migrating between two different environments or habitats, ecosystems. So the practical importance of the study actually revealed that these insects can disperse up to 2 kilometers, and they can feed other hosts, such as mostly non-grain hosts, which are in the alternate habitats. And they also can migrate into the agricultural sites or storage sites. So our current pest management is only focusing on the storage environment. We really have to now expand in the future management efforts, which need to be directing towards these appropriate breeding sites or overwintering sites far, far from the storage environment, if we wanted to have a better management program. So with that, I'm going to switch gears to another model organism, which I'm going to move into a processing, warehousing, and to the transportation system up to the grocery stores and kitchen. So this insect, Lasiodoma sericone, is also known as a cigarette beetle, very interesting beetle. And I'm going to talk a little bit about their life history parameters, their mass trapping techniques for host preference, and based on that, the mass trapping, and basically talk a little bit about mating disruption and mating delays. So as I said, this beetle is a cosmopolitan in distribution. It's a generalist feeder. It occurs in everywhere. It feeds on grain-based products. It feeds on dried fruits and nuts. Of course, it feeds on tobacco. That's why it gets the name cigarette beetle. Also feeds on all these spices we used in the condiments we used and dried medicinal products. So the larvae is the only feeding damage causing the larval instars are the one which are causing the damage to the commodities. Adults do not feed at all. They are very short-lived, just like a moth, and they actively fly to seek for oviposition sites and lay eggs and die. So our first study was to look into the life history parameters. What are their most preferred hosts and how do they lay eggs and their fecundity and development. Again, I just want to highlight here only a few of the slides from that study. We selected some of these spices, chili, paprika, cayenne pepper. We selected different grades of tobacco. NOW is the meal made out of almonds, very rich of lipids or the fat. And then, of course, our good old beet is the common one we used in the laboratory for rearing this insect as well. So oviposition is studied, and then the fecundity, you can see that wheat was the most preferred host. Very low number of eggs were laid on the cigar tobacco. That's because if you know about cigar, that's kind of like a specific kind of curing done for tobacco. A lot of acid in that tobacco when compared to other types of tobacco. So insects don't prefer to feed on that. And then egg to adult survival, again, our good old wheat remained very preferred host. Rest of them performed okay. Navel orangeworm, that's basically the NOW almond meal weight. That's basically the one which is least preferred for the development of this insect. But what is interesting is when we look into the adult body weight and longevity, of course, the wheat is the most preferred. The almond meal, which was not preferred for laying eggs or developing, however, those adults which are whoever survived on that, they basically had very heavy body weight and they lived longer. So concluding that study, the ideal medium, of course, is for egg laying, larval diet, egg survival, everything is for this is the wheat flour. And the worst performing host was our almond meal, which is very oily and not good for the egg laying or egg survival, but actually the heavy body weight was found mainly because of this fat or lipid, and also less competition. Only very few eggs are there hatched, and then that had made sure that a lot of food is there. But what is my conclusion out of this study is, don't deceive by a least preferred host, because here only a few adults survive with very heavy body weight and long lived. So those can be actually reproductively strong adults. So overall fecundity on a given female is the function of its female's body weight and its life. So they can produce more eggs, even though it is not a preferred oviposition medium. And why we did this study? Because we wanted to understand what are the different hosts preferred for this insect in order to develop the next study, which is basically the mass trapping. So again, with mass trapping, based on the previous study, now mass trapping was actually possible mainly because of the nature of this insect, because this female produced a sex pheromone. It's a blend of two compounds, cericonin and anhydrocericonin. And of course, you know that pheromone can be used for many, it has many applications in pest management, for sampling and monitoring purposes, of course, can be used for mass trapping, attract and kill, and mating disruption. So our next preface was to look into the mass trapping. So when we trap, this is a pheromone produced by female to attract the male. So if you use this pheromone on a trap, it's only going to attract the males. So let's imagine a situation. We put traps in the warehouses and we caught about 90% of the males, right? So they are not going to facilitate the reproduction. About 10% escaped. What are they going to do? They are going to survive to mate with the existing females and reproduce. So the females, the population is going to go. This is really not the mass trapping where we are completely eliminating the population. What is mass trapping should be almost you have to catch 100% of males, and if possible, also trap females. So the trap which is using pheromone only is only going to attract males. Now we have to come up with how to attract females to increase the trap catch efficiency. So the objective is to find out whether we can develop a host order-borne attract, and that can be incorporated along with the sex pheromone to reinforce the trap catch efficiency for both sexes. So this is the principle that behind the mass trapping. So again, 17 different hosts we screened because we wanted to see the response index of these beetles. The mean response index was calculated based on a two-choice walking passive air bioassay. And you can see a host of different, a suite of different hosts we tested here. And of them, of course, the chili and paprika had a highest attraction. Wheat and tobacco kind of like medium attraction, and oats or oil type of one had the lowest attraction. And if you are developing a host order-borne attractant, you don't want to work with 17 different hosts, you want to narrow down. So based on this mean response index, we narrow down our hosts to the experiments to the next level. And the next experiment was to extract the components and see how they are actually responding. So we basically selected the types of spices like chili, pepper, and paprika. They are responding very well. And then, of course, the tobacco was doing very good. And as a control or some comparison studies, we included wheat and almond oil, almond bait. So we extracted the volatiles, and again, with that volatile impregnated into these filter papers, the two-choice walking passive air bioassay was done, and then we calculated the mean response index. Obviously, these three colors represent the different types of solvent used to extract them. Some of them are the non-polar solvents or polar solvents, or kind of like medium polar solvent combination. And regardless of the type of solvent, the different types of chili, paprika, or cayenne pepper, all of them gave very high response index. For tobaccos, the medium type of polar solvent extracted the most attractive material because that's the one gave highest response index. And this actually gave us a little bit more further information about what type of host we should be selecting to narrow it down to further studies. We also wanted to see how the virgin males, virgin females, and mated females are responding to these different extracts. So the same extracts were tested for different type of, different sexes and their mating status. And you can see the purple bars are for male. Of course, they are attracted, but not as much as females. Like females are highly attracted to these hosts. And obviously the yellow bar, which is the mated female, she's very attracted to the host because she really wants to lay eggs and she's actively seeking for the oviposition medium. So I'm not going to go into the whole chemistry of it, but our next step was we selected chili peppers as our final host for fine-tuning the experiment, and a lot of different headspace collections and elutions and using silica gels, reverse phase column, and different solvents. And we tested all of them to the response index of the beetles, especially with mated females and how they are responding. And we just to show you here with different fractionation, our methanol fraction, which gave the highest response, and that is the one leading finally to develop onto a pheromone for the trap. So all this worked very beautifully in the lab. Okay, so really, do they work in the field? So that was our final study to find out whether do they really work in the field situation. So a real field study was done where in an infested facility, we selected different types of traps placed with a blank trap or just the solvent used for extraction, hexane, pheromone alone, raw chili powder, the extract of the chili, or the combination treatments, basically like the pheromone plus chili powder or pheromone plus that extract. And you can see regardless of the, whether it is combination or chili, they equally gave kind of like highest trap catchers, mean number of catchers are very high. And if you really compare the pheromone alone, we significantly high number were caught in the combination treatments because we are catching females, which was later really in the laboratory, we dissected to make sure that we indeed caught female with, spermatophore or mated females. So of course, this is the study with the mating, the mass trapping. And then now I wanted to talk a little bit about the mating disruption for the same insect. Now, what is mating disruption? So basically the female produces a pheromone. That natural plume goes in the wind and male recognizes that and he will fly to identify the female for mating purposes. So if you can disrupt this communication, how do we do that? So that is what is the principle behind mating disruption. So in the natural environment, female produces a plume, but in the mating disruption, we artificially purge the environment with very high concentration of pheromone that also emit a lot of pheromone plume. So we're creating a very heavy cloud of pheromone. This male become very confused, following to the false trails and unable to mate. So finally, what happens is either mating get disruption or mating get delayed, and finally the female cannot have the fully loaded eggs, which can be laid when if it is actually completely disrupted. So these studies were done in various different locations in South Carolina, just to show you that there are, of course, feed mills, flour mills, warehouses, even some transportation like trucks. So we initially did a lot of pre-monitoring for nearly a year and a half in order to, find out whether it's, we have indeed a good population to study. And then we actually did mating disruption in selected facilities. Other facilities were basically put into as a control. And, then we completely, post-treatment studies were done for nearly a year and a half to collect data. So these trials were repeated twice and almost 6 years or 6.5 years of studies. I'm just going to actually summarize that in 2-3 slides. So Here you can see just the treated males. We can just comparison 2 weeks before treatment and 2 weeks after treatment. What is the treatment? We are actually putting dispensers of mating these high concentration pheromone. Before the treatment, in some of those males, we caught significantly high number of mean number of beetles, but soon after treatment, you can see a trap shutdown. And that was even found in, consistently in other areas, as we also had controlled treatments. And we also compared long-term, 8 weeks before and after, and same pattern, you can see significant reduction in the number of beetles caught after the deploying the mating disruption dispensers. So this could be two phenomena. Either the male is not orienting towards the traps because they are highly confused, or maybe because we are not catching enough insects, because now the new generation is not coming enough because they are not mated. So you could see the pattern here. Sometimes it's just like goes over, as I said, it's a 6 years, and I'm just only showing a portion of it. And it's so what you can see also, the population, when we engage the mating disruption in the year 1, when we start in year 2, basically you can see a very low number of existing population. And when it goes over the time, it's so beautiful to see that the population is almost near 0. So it's like a long time of over how the effect of this mating disruption can exert. So there was a significant trap shutdown we saw, and we also did some oviposition cups where it has showed some decreased population emerging out of those cups soon after the mating disruption was engaged. And year 1 and year 2 comparison, the subsequent population is lowered from the starting population of year 1. That's the proof of that indeed the treatment is working. And this one has a greater potential to be used in the cigarette beetle population management. So the mating disruption actually shed some light on further thinking and to find out the mode of action. Is there really, because it's a short-lived beetle, and is there really completely mating disruption is taking place, or is there a phenomena of mating delay is happening? So, a mating delay is a technique which is also successfully used in controlling many insect population in other area in agriculture, like orchards and vegetable crops. Cigarette beetles are basically a short-lived beetle. So if they have to mate soon after they emerge, and if it is getting delayed, and if they mate in the latter part of their life, what is going to happen is the real hypothesis for this study, because delay, our assumption was the delay in mating can actually induce negative effects on the fecundity. And that could have the potential in use of this management. So this is basically the fitness test and the personality test. And what always reminds me is just like basically this popular song by Kelly Clarkson's, what doesn't kill you makes you stronger, right? So basically this was proven indeed in this test of some of these laboratory control tests. We basically mimic the mating disruption environment in the laboratory by artificially delaying their mating. So we let the beetles to emerge, and males and females were mated soon after emergence, and they were kept together or they were separated after 48 hours. And then we control their life, like basically control the age of mating. The day 1, they were separated and allowed to mate forever, or 2, 3, 4, up to 14 days of delaying. And then looking into their longevity, their basically progeny production, how that could be affected by this kind of delayed mating. And what is so beautifully shown in this comparing the longevity studies is the virgin female who got never mated lived significantly longer, 31 days, when compared to a mated female who only lived about 19 days. And this phenomena was same to the male as well. The virgin male who never mated lived about 30 days when compared to a mated male. And this dark blue lines standing out are the one which were virgin males or females. So what is basically concluded is if they mated, they're basically converting their, spending their ATP or the biological energy into investing into this egg production. The amount of energy available for the longevity is reduced because they don't feed as an adult, so they don't consume new energy. Therefore, there is a differential fitness cost for them to be mated. That means if the energy is spent for producing eggs and they lived shorter, but if they never mated, then that energy is preserved for long life. And some other studies we also did in order to see the effect of what if both sexes were delayed or one of them were delayed, either males were delayed or females were delayed, what would be the effect? And then we looked into the F1 generation, the progeny production. And it's also interesting to see there when both sexes were delayed or females were delayed, as expected, similar effects, right? And when males were delayed, the effects were slightly different from the previous two. And what is so interesting also to note, soon after emergence, if they mated, they didn't lay a lot of eggs actually, because they are probably not sexually mature on the 0 hours from emergence, right? But 2 to 3 days of emergence, they kind of mated, they laid significantly high number of eggs, so the progeny development is high. And then if the mating is delayed, you can see up to 6 days, the progeny production decreased and it's kind of plateaued, right? So this study indeed shows that the critical period for mating for cigarette beetle is about 1 to 2 days of the emergence. If we can disrupt this kind of mating, obviously we can influence the amount of progeny produced for actually contributing to the next generation. So the conclusions are basically unmated adults lived significantly longer when compared to the mated adults. And mated females also lived significantly longer than the mated males. And the progeny production basically decreased with the age of the mating. So longer the wait, the number of F1 generation is reduced. And delayed mating has a significant effect on the females, actually significant effect on females. And it's reduced the reproductive performance in cigarette beetle. In the last few minutes, I just want to touch base on this international work with the USAID. It's basically an assessment of post-harvest losses for to establishing a partnership to reduce the post-harvest losses in countries such as like Ethiopia. So this was really a consortium of a larger grant led by Kansas State University, and there are some other, partnering institutions here. You can see them, and I am representing South Carolina State University. It was funded by this USAID program for Feed the Future program, which is basically to secure the global hunger and food security research strategy. The research grant was funded for 4 countries: Bangladesh, Ethiopia, Ghana, and Guatemala. I actually co-led the team for Ethiopia. So we have been working since 2014 January. So it's almost like now, 9 years of work. And if you're not sure about Ethiopia, it's just like now a lot in the news these days with the ethnic cleansing happening in that country. It's a country with a lot of fertile land, very diverse climate, a lot of rainfall, and it's still very traditional agriculture. You could see this is basically called the pit storage or kind of underground storage. This is how mostly grain is stored. They dig a hole and, put a tarp inside and then basically store the grain and make a clay lid or something, cover it and plant a tree on top of it so nobody knows grain is underneath, so no theft, because that's a serious problem. whenever they need it, they go into that and take the grain out. Sometimes they do hunting and use the animal hide, such as like these bags are made to store inside of the house, or mud huts like this. Very, very foolproof. Birds and rodents and insects can easily enter into these kind of structures. And just to show you some of these damages which we encountered, like in a flour mill, you can see how the red flour beetle are gorging on this stored flour. These losses can account up to 60% in that country. These are some physical damages in a wheat warehouse, a lot of torn bags, and we are basically walking on a carpet of wheat, and a lot of pigeons and other birds and insects damaged, rodents. You can also see how it is infested with bird fecal matter here. And this is a sample I'm actually showing you from the pit we took out from the sorghum, and you could see it's a lot of mycotoxin contamination, insect infestation. There are insects, born into this hole here, you can see. And this is basically what the people are feeding, because it's once a year they produce this crop, and they don't have anything else for access to. So our program is a two-pronged approach. First of all, to undertake applied research, basic applied research in order to mitigate the losses of that 60% to lower levels, and all possible methods. We had a very diverse team of engineers and pathologists and entomologists, economists, gender specialists and nutritionists working towards this program. And also create infrastructure capabilities, not the idea of introducing expensive techniques for them, but to improve the on-the-shelf technology, what they had, in order to minimize these losses. And also to develop and establish technology-enhanced engagement capabilities. So just to highlight, the 9 years work has made tremendous progress, and we have created awareness. We worked with many universities, Department of Agriculture, the non-governmental organization, the Africa Rising, so many different areas we reached out, including farmers, and several good things have happened due to this program. So with that, in closing, I think my time is approaching here to finish up. So just want to highlight that insect problems are still elephant in the room. And in stored products, prevention is the #1 priority. We just have to prevent the problem before it occurs, because if the problem occurs, we cannot compensate, because these are the harvested product and it's not going to grow back again. If insects are encountered, make sure to identify them and identify the root cause of the problem. Where are they coming from? Are there any alternative sources? And how can we really target a bigger IPM program area? And insect management is just like not just spraying and forgetting about it. We really have to think about it in a bigger picture, like think outside of the box. And if we are practicing a good prevention program and incorporating all the other methods in the tools which are available in the toolbox, then fumigation could be the last choice once all our other measures are failed. I want to give a shout out to all my students, postdocs, everybody who has numerously contributed to this work, and without them, I could have not done with any of these. And I just can't go with the names, so I'm just showing you the pictures, and all my collaborators, funding agencies, especially with Kansas State University, Clemson, the USDA-ARS in Charleston, Oklahoma State University, the USAID grant, which is basically the, coming through Kansas State, and my Fulbright Scholar program through the University of Peradeniya in Sri Lanka. And finally, I also want to thank you for your patience to listening to this. And if you have any questions, I'm really happy to answer.
Great, thank you, Rosanna. That's a great talk. So questions. I have a lot of questions. I'll wait for later. Maybe somebody else can ask first. Well, if not, I want to ask you, Rosanna.
Yeah.
I remember Purdue University have a, improved storage bag called the PICS bag, right? Are they using it?
Yes, one of our trial is basically to test all the different vacuum packing techniques or the controlled atmospheric technique to store. So we tested in Ethiopia the PICS bag, and there are Grain Pro bags. It's a different company. We also had some kind of different products available in Africa. We tested. So of all the products, the PICS bag is the best. I would say that. Yeah.
But are they affordable and can they be widely used?
Yes, the affordability is about 20 birr for the bag, which was almost closer to they are selling, I think it's subsidized rate about $1.50. But at that time, it fluctuates now because I haven't been to Ethiopia since the COVID and now the conflict in the country. So it's almost 2 years, but it's about now 40 birr. So that's, and also it's multiple use. So the farmer can use it for 2 to 3 times.
Okay. Kasha.
Hi, yeah, I just had a question. Thank you for your talk, by the way. But my question was, so let's say you have bags that are infested, right? Would then the buyer of those bags just take the whole load and say that that load is not accepted anymore? Or like, how do you screen for which bag could be infested, which bag is not infested?
So that's a good question. In the United States, what they do is basically the grain is sold from farmer's bin to the elevator or the country elevator or terminal elevator. And when the elevator managers, usually when the farmer brings the grain, they sample it. And in the US, we don't bag it. They bring it on a truckload and they dump it, and then they take the sample. And there are certain criteria. For example, 1 kilogram has 32 insects or so many insect fragments, which is already set there. But if you go to a foreign country like in Ethiopia, well, they are mostly bagged, and because they are in the bag and you have these augers, you have to make basically poke through that bag and take the sample and see and whether there are any insects existing. And their level of infestation allowed on the grain could be different because it's different from country to country. For example, in US, we allow 32 insects, but in Australia, 0 tolerance. You cannot have any insects. So it's different from country to country. But also, when you have a bag and you are damaging it, you are basically making another entryway to go into for other insects to invade to that.
Okay, thank you.
You're welcome.
Rosanna, so in the oviposition cups you used for evaluating the mating disruption on the egg production, how do you know the reduced progeny is due to mating disruption or due to the population decreased? Do we have a control for that?
Well, we have control, but it's not within the facility. So the control is not the homogeneous control. So for example, mill 1 is where the mating disruption was done. Mill 2 is not mating disruption done. And then what we did is we evaluated the oviposition cups. I mean, you set the oviposition cups before you are setting up your dispensers. For nearly 6 months, and you are looking the population. And then you, on your treated facility, you put the mating disruption dispenser, but in your control facility, you don't have it. So you continuously monitor. And then you look for the post-treatment oviposition. And then you bring it back and you incubate it in the laboratory and give for 1 month and then emerge, and then you count the adults coming out. And then compare before and after. Yeah.
Okay. So you also said, after placing the dispensers, then you have a trap shutdown, right? So how do you separate this trap shutdown and the population reduction? Because the lower number of trap catch can be both reasons.
That's a really good question and observation. As I mentioned, the trap shutdown doesn't guarantee that mating disruption is happening, because trap shutdown, the trap is only you're using a monitoring pheromone in that, and maybe males are confused and they are not, orienting towards that trap because they are confused, and that doesn't guarantee that the mating disruption is happening. How can you prove the mating disruption is over time? You look into the population and looking into the oviposition cups, and then over time, long time period, you trap, and that's when you can conclude whether there is an effect on this or not.
Okay.
The immediate trap shutdown is just an indicator that they are confused, but it doesn't guarantee that mating disruption is happening.
Right.
Siadan?
Yeah.
Okay. Hi, this is Xiaodan. Hi, Dr. Mahroof. It's great talk. It's great talk. I know you did a lot of work, but today I feel so fascinating. You did a lot. Yeah, it's so great. And I have a question about the cigarette beetle. Based on your experience, is there any like the distributed pattern in other states or other places about the cigarette beetle? I had some data about the Indian mimos in the coffee bean warehouses from the pheromone traps. I also collect a lot of cigarette beetle. Actually, the cigarette beetle has more than the Indian mimos, and I didn't find any like the distributed pattern here. They just seem like randomly there.
That's depend on which facility you are talking about and what products you are talking about. So in the mills, we have certain, there is no uniform flight behavior you can really mark, but basically they go kind of like low catches during the winter months. You don't catch a lot. During summer, they are really peak, and then fall and spring kind of in between. And also certain times that could be altered, because I worked with, I don't want to name the facility, but I worked with the warehouse. And whenever they brought the products in, regardless of whether it is winter or summer, if the product is infested, then you catch more.
Oh, yeah.
So that could happen. Because, so it's sometimes if you really look into an existing population, then what happened in the laboratory, in the colonies we maintain, winter, you cannot get anything out of them. They are very kind of like, they are not really hibernating or, but it's just the production is very low. But even though it's in the laboratory control environment. But in the field, only one warehouse, not the warehouse which I was talking about, I saw that whenever they brought a new stock in, and if the stock is infested, then you catch more on the traps.
Yeah, same here. In New Jersey, based on my experience, the cigarette beetle, both the cigarette beetle and the Indian mimos came out from the early May until the end of the October. It's similar here, but the Indian mimos, you can very easily to see there are 2 to 3 peaks every year. But for the cigarette beetle, there's no distributed pattern there. And also my second question is, if there's a new incoming shipment, or there's always have new infestation of the products, how can we do about that?
That's a big problem. So that's all depending on what they usually do is when shipments come from a foreign country, in the harbor or wherever the port, they usually sample the product, and if it is heavily infested, they fumigate it before let in come in to the wherever the distribution. But still, there could be something which is, the eggs hibernating, which may not be, seen inside, or the eggs which can, hatch later after it is, gone through the supply chain. And mostly the product which I was really dealing with is coming from South America and Africa. And these are basically like, whenever they brought a big shipment in, then regardless of all the screening and all the things done, but the product coming in can bring in some infestation. So basically you have to treat them before coming in, or you have to heavily sample them, identify the insects, and if they exceed to a certain level, you treat them before bringing it to the other storage environment.
Okay, thank you. I still have some questions, but I'll ask you later.
Okay, you're welcome.
Shannon, unmute yourself first.
Thank you. Thank you for a great talk. I was actually really interested in looking at all the different phases of that, and especially implications in some countries that don't have the technologies that are available in other countries. I thought that was really interesting. The question I have is specifically on the slide referring to the 8 weeks before and 8 weeks after implementing the mating disruption. I noticed, most of what you talked about was focused on mill 1 and mill 2. And then it looked like you included a warehouse in there, but it didn't look like the warehouse had the same, over the time, did not have the same reduction as what you saw in the mill houses. Am I incorrect in that? And if I'm not, is there a reason that you think that's the case?
I actually picked only, thank you for that question, Dr. Sked, I guess, Shannon.
Shannon, yeah.
Okay, sorry. I was reluctant to pronounce that well, so anyway. Your observation is correct, but what I have shown is only merely a sample of what the long-term studies, nearly 6 years of studies. And the warehouse, I have a separate table. Basically, we brought the warehouse in after the first trial is ended. Because during the first trial, we couldn't, this is a big corporate, we couldn't get MOU and agreements done. So it took a long time to, get that access to the warehouse. So the warehouse access came only after the second trial was initiated. That's why you probably saw that it was basically, but the data for warehouse was actually summarized separately. And in the publication, you would see a table. And that table shows that there's a significant reduction. It's the same way, 2 weeks or 8 weeks after and before, there's the same effect, yes.
Great, thank you.
You're welcome.
Yeah, I think we should end now. It's already 12. So Rosanna, maybe we should have a 5 minutes break. After that, the students will continue, stay, and for more discussion, everybody else can leave. Okay, thank you, Rosanna.
Wonderful, thank you so much.
Thank you very much.
Yeah, appreciate everybody coming in today.
Okay, 5 minutes later we can come. Okay, 12:06.